Scientists have just achieved what was once thought impossible. Down syndrome affects about 1 in 700 newborns in the United States, caused by an extra copy of chromosome 21 that disrupts normal cellular function. For decades, researchers have searched for ways to address this genetic condition at its source, rather than just managing symptoms. Now, scientists from Mie University in Japan have achieved something remarkable. Using CRISPR gene editing technology, they've successfully removed the extra chromosome 21 from cells of people with Down syndrome, potentially restoring normal cellular function. The research team, led by Ryotaro Hashizume, used a technique called allele-specific editing to precisely target and cut away the surplus chromosome. This approach is groundbreaking because it addresses the root cause of Down syndrome, rather than just treating its effects. Down syndrome, also known as trisomy 21, occurs when cells carry 3 copies of chromosome 21, instead of the normal 2. This extra genetic material forces cells to overwork, disrupting how genes are expressed and proteins are manufactured throughout the body. The CRISPR-Cas9 system works like molecular scissors, designed to recognize and cut specific DNA sequences. The researchers carefully programmed these scissors to target only the unwanted 3rd chromosome, leaving the other 2 copies intact. The results were impressive. After removing the extra chromosome, the treated cells showed normalized gene expression patterns and reverted to typical protein manufacturing. The corrected cells also demonstrated better survival rates and grew faster than untreated cells. Even more encouraging, the technique worked not just on laboratory-grown stem cells, but also on mature skin cells taken directly from people with Down syndrome. This suggests the approach could potentially be applied to different cell types throughout the body. The corrected cells showed additional benefits beyond normalized gene activity. They produced fewer harmful reactive oxygen species, indicating improved cellular health and reduced biological stress that typically slows cell growth. While this breakthrough is promising, the researchers emphasize that the technique is still far from clinical use. Scientists are working to refine the method to ensure it only targets the extra chromosome, without affecting healthy genetic material. The study, published in PNAS Nexus, represents a major leap forward in genome editing capabilities, demonstrating that CRISPR can remove entire chromosomes rather than just making small genetic corrections. This research opens new possibilities for addressing genetic conditions at their fundamental level, though extensive safety testing and refinement will be needed before any potential therapies could reach patients. For more on this story, find a link to the article on the #1 science news site in the world, Earth.com, in the description.